Yes, Dr. Nidhi, please start.
Good morning to all. Hello to everyone joining today's webinar. I'm Nidhi Mittal, and as the co-convener of this event, I'm delighted to welcome a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learnings as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I'm confident that today's session will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome everyone. I look forward to a productive experience for all participants. Thank you for joining us. We are gathered today for the 72nd SPSR webinar on eyeing the future platforms for enhanced ocular therapeutics, a distinguished event in the series hosted by the Society of Pharmaceutical Sciences and Research under the esteemed guidance of Dr. Shashi Alo, National President of SPSR. The society continues to play a pivotal role in fostering innovation and advancement in the field of the pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of these strategies. Our mission is to organize conferences that support pharmacy related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving toward these objectives, we aim to elevate the quality and impact of the pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Professor Preeti K. Suresh, Director, Malaviya Mission Teacher Training Center, and Professor, University Institute of Pharmacy, Pandit Ravishankar Shukla University, Raipur, Chhattisgarh, India, as today's distinguished speaker. Professor Preeti K. Suresh expertise greatly contributes to our event, providing substantial knowledge and insights. Her participation is certain to enrich our discussions and expand exploration of the eyeing the future platforms for enhanced ocular therapeutics. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serve as the initial secretary of SPSR and the managing editor of both IJPSR and IJP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. I'm pleased to invite Mrs. Monika Sabharwal to warmly welcome our distinguished guest speaker, Ma'am Professor Preeti K. Suresh. Over to you, ma'am.
Thank you, Nidhi Mittal. Hello, everyone. I am Monika Sabharwal, the convener of today's 72nd SPSR webinar, and I am delighted to welcome you all on the behalf of Society of Pharmaceutical Sciences and Research SPSR. Today's webinar is dedicated to explore the exciting frontier in the healthcare, eyeing the future platforms for enhanced ocular therapeutics. Our topics highlights the innovative therapeutic platforms focused on advancing the eye care, an area that is rapidly evolving and holds an immense potential. We are honored to have with us an esteemed expert and today's speaker, Professor Preeti K. Suresh. Ma'am, Professor Preeti K. Suresh is a director of Malaviya Mission Teacher Training Center and professor at the University Institute of Pharmacy, Pandit Ravishankar Shukla University in Raipur, Chhattisgarh, India. With her deep knowledge and distinguished career in the pharmaceutical sciences, Professor Preeti K. Suresh ma'am will provide invaluable insights into the future of ocular therapeutics and exciting advances that are shaping this particular field. This event has attracted an overwhelming response, with more than 2,000 registrations from 20 countries, reflecting the global interest in the cut edge ocular therapeutics. This is a milestone for the SPSR as we celebrate our 72nd webinar. Throughout this journey, we have been motivated and inspired by the tremendous support of our participants and by the enthusiastic ongoing feedback we always receive. Your insights encourage us to continuously raise the standards of these events, bringing the top tier professionals and the impactful topic that drive out your field forward. At SPSR, we are committed to present a diverse range of topics, often chosen on the basis of feedback and the suggestions from our participants. To make it even easier for everyone to join us, we have recently launched SPSR webinar membership program for all of us. This allows the participants to register just once for the entire year and gain uninterrupted access to all SPSR webinars, eliminating the need for the multiple registrations. You can register for this membership directly on the SPSR website. In addition, SPSR is excited to announce that nominations for the SPSR awards are now open. This is an opportunity to recognize the outstanding contributions in the field of pharmaceuticals and healthcare. You can visit our website to submit your nominations and join us in celebrating the excellence in your profession. We also encourage the collaborations with both national and international institutions. So, any organization interested in the participation with the SPSR is welcome to reach out with your proposals. Our goal is to foster a global community where we share insights, challenges, and breakthroughs, enriching the pharmacy profession for overall. None of would be possible without the dedicated SPSR team working behind the scenes, who, whose hard work and the commitment make these webinars successful every Sunday. Their efforts, along with their invaluable supports of our audience, allow us to deliver the high quality knowledge sharing sessions that positively impact the global pharmacy community. Before we commence, I would like to warmly welcome Dr. Shashi Alu, our National President, whose visionary leadership has been crucial in the impressive transformation of the SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Nidhi Mittal, the co-convener of this webinar, and organizing committee members, Dr. Padmani Shukla, Mrs. Swati Jain, Dr. Saurabh Kose, Dr. Sanjay Nagdev, Dr. Pradeep Gulani, Mr. Ankur Agarwal, Mr. Junaid Tantre, Dr. Payal Mittal, and Dr. Khushbu Mishra. Their tireless efforts have played a crucial role in transforming the vision of this event into a reality. We thank you for each of you for joining us today and your continued support, which has helped the SPSR community grow and thrive. We are confident that today's session will deepen your understanding of this particular topic. Join the SPSR vibrant community of over 30,000 pharma professionals actively engaging across the LinkedIn, YouTube, Facebook, and WhatsApp. Connect, learn, and grow with the top tier webinars and the exclusive content. Do not miss out this opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSI YouTube channel and enabling the notification bell icon for instant alerts. Follow our journey in advancing the pharmaceutical sciences that will benefit the society. Once again, thank you for being here today. We hope today's session will provide a valuable insights and inspiration. Now, without further ado, it is my pleasure to welcome our distinguished speaker, Professor Preeti K. Suresh ma'am, who will lead us through this very fascinating topic. Over to you, Nidhi Mittal.
Thank you, ma'am. I appreciate Mrs. Monika Sabharwal granting me the opportunity to present this webinar. And now we will start with a short introduction to our today's guest speaker, Professor Preeti K. Suresh, ma'am. Uh, Professor Preeti K. Suresh, she is an accomplished academic and researcher with over 25 years of the experience in the fields of the pharmaceutics and drug delivery. Currently, she is a professor of pharmaceutics at the University Institute of Pharmacy, Pandit Ravishankar Shukla University, Raipur, and serves as the director of the Malaviya Mission Teaching Training Center, formerly known as the Human Resource Development Center at the same university. She completed her bachelor's, master's, and doctoral degree in pharmacy from the Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur University, Sagar. Professor Preeti was awarded the prestigious International Foundation Research Fellowship in Japan in 2005, which allowed her to conduct postdoctoral research at the university in Kobe, Japan. Her work primarily focuses on nano carriers for drug delivery applications, where she has made significant contributions with over 60 scientific publications in indexed and peer-reviewed journals and more than 100 papers presented at national and international conferences, seminar, and symposia. Additionally, she has authored 8 book chapters and has written various popular articles on health and women's issues. In addition to her teaching and research, Professor Preeti has held various administrative roles, including Dean of the Faculty of Technology and Director of the University Institute of the Pharmacy. She is actively involved in reviewing the renowned international journals on advanced materials, nanotechnology, nanomedicines, and drug delivery. She also serves on academic committees for different governing bodies and universities, contributing significantly to the advancement of the pharmaceutical sciences. She is a life member of IPA, Indian Pharmaceutical Association, and APTI, Association of Pharmaceutical Teachers of India. She also contributes to the editorial board of the APTI Women's Forum newsletter and is a member of the state level task force for implementing the National Education Policy 2020 in Chhattisgarh. With her wealth of experience, dedicated to research, and commitment to educational advancement, Professor Preeti K. Suresh is a respected figure in the field of the pharmaceutical sciences. Now, I want to inform you with great satisfaction and pride that we have received more than 2,000 registrations for this today's webinar. Registrations received from the India, Bangladesh, Canada, Egypt, Indonesia, Iran, Jamaica, Kenya, Kuwait, Malaysia, Maldives, Nepal, Nigeria, Oman, Pakistan, Philippines, Saudi Arabia, Thailand, Turkey, and United Kingdom. We are deeply moved by the substantial and varied responses we have gathered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker for today's webinar, Professor Preeti K. Suresh ma'am with us. Welcome, ma'am.
Thank you. Thank you very much, Dr. Nidhi, for a very kind and humbling introduction. First and foremost, I would like to thank Dr. Shashi Alok from the Society of Pharmaceutical Sciences and Research for giving me this opportunity to come on this platform and share some of my experiences and insights on the topic that we are going to discuss today, and also to Dr. Monika Sabharwal for her very proactive approach in further enriching our field of pharmaceutical sciences. So I will just start sharing my screen. I hope I am both audible and visible to all.
Yes, ma'am.
Yes. So I hope my slide is visible, right? Yes, Dr. Nidhi, can you just confirm?
Yes, ma'am.
Yes, okay.
It's visible. Yes, ma'am.
Yes, okay. So, in today's session, we are going to discuss on what are the various platforms for enhancing the ocular therapeutics. And with that backdrop, I would also like to discuss with you what are the major challenges that are ahead, especially when we are dealing with the ocular therapeutics. As you all know, eye is one of the very crucial or very important vital senses, and 80% of all our learning comes through the eyes. So there is no need to emphasize on the importance or significance of this sense organ, and any disorder or any disease which is going to affect the eye is naturally going to be very alarming for any individual who is affected by this disease or disorder. And it is very important for us to understand that eye is a very complicated organ, although it is very small in size, but you can say it is, in respect to complexity, it is second only to the brain. And although it is a very small organ, it has very various parts, various which have to work in perfect harmony, and only then we are going to enjoy the benefits of what sight brings to us. But before proceeding further, I would like to draw your attention to this quote, very deep quote by F. Scott Fitzgerald, who very noted that the test of a first-rate intelligence is our ability to hold two opposed ideas in the mind at the same time and still retain the ability to function. One should, for example, be able to see that things are hopeless and yet be determined to make them otherwise. Why I am sharing this quote is because we have to understand that sometimes whatever new ideas, whatever new systems that we may be proposing, we have to move forward with that idea, with that concept, with a belief that there will be some uncertainty involved. And if something better comes along in our way, we have to adopt that new approach. Because similar is the case in the field of pharmaceutical sciences. Daily newer systems are coming up, newer understanding of the various pathologies are coming up, and we have to take in our stride whatever ambiguity will be there, and in the same way we have to navigate through the complexities and adopt the newer approaches whenever we are looking forward to finding new solutions for a problem. So in the extension of this, you can say cornea is a very, it has a duality in its structure. Duality in the sense, it is having structural simplicity, but behind that simplicity is a very mechanistic complexity. So there is a dichotomy, you can say. It is both a simple and a complex structure. And this duality, you will, as if you are going into the ocular drug delivery research, you will see this duality exists in different phases or different steps. And if you want to see the ocular diseases, as I mentioned earlier, it affects one of the most indispensable senses, that is sight. And if any point of time there is some impact, or in some way there is some glitches that is occurring in our slide, it is going to impact the quality of life, and particularly for the elderly patients. And when we are looking forward to finding solutions or devising newer formulations to treat the ocular diseases or disorders, we have to take into account that eye, it is a very delicate organ. It is a very sensitive organ. And we have to take into account various aspects whenever we are designing a formulation or a delivery system to treat the diseases which are affecting the eye. But before I go further, I would just like to draw your attention to the fact that the incidences of ocular diseases are on the rise. And certain reports have been indicative that in 2020, where the incidences of ocular diseases was 14 million, but this number will be doubling in the next 30 years. And by 2050, there will be more than 30 million patients who are affected in some way or the other by certain diseases. And the major concern or the reason for this increase in the diseases which are affecting the eye is because demographically we are moving towards an older age groups, and as the number of aging population is increasing, it is very natural that this is going to translate into increased number of patients who are in some way or the other affected by visual impairment. And with this aging population and with the increase in the lifespan, the potential of blinding eye diseases, whether it is age-related macular degeneration, or it is diabetic retinopathy, or it is glaucoma, the incidences of these diseases are going to rise. And if we just go on to see the market for the ocular drug formulations, and as I said earlier, the prevalence of disorders of the eye, both acute and chronic, these are on the rise. And the opportunities are rising for coming out with newer formulations, and that is one of the reasons why the pharmaceutical industries are also focusing too much on these sector of formulations, because this sector gives us the opportunity for customization, for providing personalized medicines. But for all this, we also have to see to it that regulatory support is also there for the innovative products that are coming in this field. Now, if we go on to see what are the major ocular diseases which are having an impact on the visual acuity, whether affecting the vision or it can also lead to blindness. Some of the major diseases which have been on the rise, these include the cataract, glaucoma, age-related macular degeneration, diabetic retinopathy is on the rise, dry eye syndrome that is also on the rise, and allergic conjunctivitis. And if we see the global market that is existing for the ophthalmic products, these are also on the rise. And this market is estimated to grow at a rate of 7.4% in the coming 6 years. And also we have to see to it that major of the products that are ophthalmic products that are existing in the market, these are the conventional eye drops, because eye drops, these are more convenient for the patients. These are more adaptable to self-administration. But other products like eye solutions or suspensions, they also constitute a major share in the global market. Then ointments are also there, and newer gels are coming up. And also the systemic administration of the drugs for ophthalmic diseases, whether it is in the terms of capsules or tablets, these also constitute a significant sector of the ophthalmic drug market. Now, one of the major challenges whenever we are talking about the ocular formulations is the poor bioavailability issues, because the poor effective drugs are there, but the major constraint that limits the therapeutic outcomes is the poor bioavailability of the products when we are administering them through the uh, local ocular routes. So the major issues which constitute to the poor bioavailability, they are twofold. One is the basic protective mechanism that is existing in the eye. Being a sense organ, physiologically it is designed to respond in such a way that the whatever any foreign material that is being introduced into the eye, that has a very short residence time. And these mechanisms like your blinking, reflex, lacrimation, and the nasolacrimal drainage, these are the protective mechanisms of the eye which contribute to the poor bioavailability of the drug. Another are the anatomical barriers which are existing in the eye, whether it is in terms of lipophilic corneal epithelium, or it is the hydrophilic corneal barrier, or the scleral stroma, or the conjunctival lymphatics, whether it is choroidal vasculature, and the various blood ocular barriers that are existing. And all these barriers, there is an interplay of these barriers, and sometimes the bioavailability of the drug when they are administered through the ocular route are as low as 1%. Even achieving 5% is a very ambitious target when we are looking forward to the ocular therapeutics. So some of the major barriers, if you see, these are the major barriers which are existing, and these barriers are in terms of the tear film barrier. Then the corneal barrier is also one of very significant barrier. Then conjunctival barriers are there. Then blood aqueous barriers are there, and then blood retinal barriers are there. So all these barriers, even though, as I mentioned earlier, eye is a very small organ, but the number of barriers that are existing that presents a very challenging target for circumvent all these barriers and reach to the site of delivery. And specifically, when we are talking about the posterior segment diseases, that is all the most challenging. So we can understand that there is a significant market is existing for coming out for products for reaching to the posterior segment diseases. So when we are talking about the ocular delivery systems, we have to take into account that this particular class of products, there are multiple dichotomies are existing. There are both opportunities and challenges, and we have to merge these multiple dichotomies because we may have to take into account both the acute versus chronic diseases. We may have to take into account epithelial versus endothelial damage. We have to take into account the fibrotic and the vascular pathologies, the chemical exposure versus what are the various counter measures that are to be taken. We also may have to take into account whether we have to use that approach for monotherapy or we are going to use a dual therapy or a combination therapy, or whether we are going to establish the efficacy of these products using animal models or the human models. So all these aspects have to be taken into account whenever we are venturing into designing a product for the ophthalmic disease. So if we go on to see the ophthalmic drug delivery has been lagging behind, you can say. It is not because there is lack of innovation, but the major challenge has been that we have to reach out to the eye effectively. That is the foremost challenge. Drugs are existing, but how effectively we can target those drugs to the eye, that is one of the major challenges. And example, even if we are looking for designing a eye drop, it is very important to understand that even designing an effective as simple formulation as an eye drop, which will be safe in the eye, which can reach the target area in the eye, and which is able to retain its stability in its container, that itself is a big achievement. So you can just understand when we are venturing into more complex or more complicated formulations, the challenges are multifold, right? So now from the conventional as the major market, you can say, of the ocular formulations, they are dominated by the eye drops and the injections, but now gradually we are moving towards more drug delivery innovations. So some of the major challenges you can say which are associated with the ocular drug delivery is whenever we are these conventional or even some newer formulations are being developed, and when we are using these different routes for reaching to the various targets for the eye, they have their own set of limitations. For example, if we are looking forward to some topical formulations, the major constraint is the limited penetration, which is as less than 5%, and the continuous lacrimal secretion that leads to the washout of the drug from the site of absorption. So the retention of the formulation once it is topically instilled is very low. And this leads to the need for multiple instillations which is not convenient from the point of view of the patient, and it ultimately translates to poor patient compliance. Another approach or route of administration is the systemic administration. Sometimes, when we have no other option of designing or administering the drug through the topical routes or through the various injectable routes, we have to go for the systemic administration. But this has its own limitations because, due to the existence of the various ocular barriers, and this can lead to limited and variable penetration of the drug, and also the incidences of systemic toxicity is also high, because if we want to achieve a significant concentration of the drug at the ocular sites, the other sites in the body will be exposed to a much higher concentration of the drug, which may be responsible for the systemic side effects and even lead to toxicity. Even during the last many years, many intraocular implants have been designed, and but with these intraocular implants, there is the risk of retinal detachment, and also chances of intravitreal hemorrhage is also there, and it is also an invasive approach. Then intravitreal injections have been also being used specifically for targeting the drugs for posterior segment diseases, where the risk of retinal detachment, it is an invasive route, and there is a risk of retinal detachment, risk of hemorrhage is there. There is the chances of endophthalmitis and the formation of cataracts, and it is also rapidly diluted by the fluids, and also sometimes the repeated procedures may be necessary, which becomes cumbersome because it is an invasive route. So if we go on to see what are the major challenges in ocular therapeutics, the major challenges is there is no cure for many ocular diseases, whether we are talking about dry age-related macular degeneration or diabetic retinopathy or glaucoma. And another issue is the whatever drugs are being administered for ophthalmic diseases, these are not based on the unique characteristics of ocular diseases, but these applies for agents which are developed for non-ocular diseases to the eye. So whatever drugs we are seeing for the treatment of for the ocular diseases, these are not specifically drugs which are being developed for ocular diseases, but these were drugs which were synthesized or developed for other non-ocular diseases, but now they are being adapted for the eye. So they are not being developed with the taking into account the pathophysiological conditions that prevail in the ocular diseases. Another major issue is that the ocular diseases are, most of the ocular diseases, these are multifactorial diseases. Multifactorial diseases in the sense there are no single factor is involved for those diseases. There may be a number of genetic factors, number of environmental risk factors which contribute to the development of those disease. And sometimes one drug that may only work on a subset of patients only, because they will be active only against some only one or two factors. So the whenever we are looking for a product that should be efficacious, that should have good therapeutic outcomes in a larger set of patients, so that becomes a major challenge. And targeting to the posterior segment diseases it remains a challenge. We have to rely only on the invasive approach for reaching out to the posterior segment of the eye. So if we go on to see what are the various clinical needs, why we need to improve the ocular delivery, one is due to the inherent lower bioavailability, lesser retention time of the drug, we have to go for the frequent administrations, which is not at all convenient from the point of view of the patients. And if the patient's convenience is not taken into account, we all can very well understand that the patient compliance is also reduced. And whatever newer formulations are required, that have to be free of risk in the terms of infections or any retinal detachment. Then the burden of treatment also have to be reduced. Then the conventional formulations that we are talking about, they are not able to achieve a constant drug concentrations. There is a very fluctuations in the drug levels, what we call it as the peak and troughs at the drug levels. So sometimes the better therapeutic outcomes cannot be achieved due to these fluctuating drug levels. And why we have to focus on delivery? Because ultimately we want continuous and controlled drug release. We have to improve the efficacy and lower the toxicity, and we have to improve the compliance of the patient so that the treatment burden can be reduced. So what are the future, what does future holds for us, or where we are looking forward to? We can be very optimistic. We have, because considerable research is underway, and during the next few years we will see there will be more drugs will be coming up for conditions like age-related macular degeneration or glaucoma, and these drugs will be offered via sustained release mechanisms. So whenever we look forward, or we are going to design a formulation, any form of drug delivery systems we may be looking forward to, but there are certain criterias that we have to take into account. What we have to see to it, whether what is the nature of the drug, whether the drug is hydrophilic or lipophilic. Certainly lipophilic drug will be more preferred because it will be more readily able to cross the biological membranes. Then if the drug is, for example, if the drug is non-ionizable and it is lipophilic, the drug is more liable to concentrate in the corneal epithelium. But if the drug is ionizable and lipophilic, it will more readily partition into the aqueous humor. So we have to take into account what are the properties of the drug, and in that way we can design a optimum formulation. Then we have to see to it what is the type of carrier that we are looking forward to, what is the vector type. So all this depends upon what are the drugs we are looking forward to for delivering to the various segments of the eye. So whatever carrier that we are selecting, we have to make sure that it has a high loading dose, because if we are looking forward to a formulation which has to be instilled into the eye, it cannot be more than 1 drop. So we have to be very sure that the dose of the drug is carried in 1 drop, and that is only possible when the loading drug has to be potent enough, and the loading dose also has to be very high. Then what is the size of the carrier? So size of the carrier must be as low as possible, because size plays a very significant role in the corneal uptake and what the passage that the drug or the carrier follows. Then we also have to take into account the osmotic pressure, because the whatever formulations that we are instilling into the eye, it has to be isotonic with the physiological fluids, because if it is not isotonic, it will cause irritation, it will cause lacrimation, because the first defense mechanism of the eye is the enhanced tear formation, so as to remove the any foreign object that is coming into the eye. So the formulation has to be isotonic in nature, and the pH of the formulation has to be as close as to the physiological pH to avoid any irritation or the lacrimation. And sometimes we may have to use certain buffers, and if buffers are to be used, we have to take care that we have to use lowest possible buffer concentration, and it should be not more than 0.1 molar. So these are some of the uh, criterias that we have to keep in mind while we are designing a ophthalmic drug delivery system. Now, there is so much talk about the nano formulations these days. Nano formulations are the buzzword, you can say. And if you ask me whether nanomedicine is useful for ocular diseases, the answer is certainly yes. It is very useful for the ocular diseases. Where it is useful? It is useful where cellular penetration is required, where we desire that the carrier should enter into the various cellular sites. Or it is also useful where we require that optical clarity is maintained. It is important that whatever formulations that you are instilling into the eye, it should not be opaque in nature, it should not interfere with the visual acuity, or it should not hinder the uh, sight. So it is very important that whenever we require optical clarity, nanomedicine certainly carries with them that feature. But it is also important for us to understand that the drug loading has to be very high. Whatever carrier that we are using, the drug loading efficiency should be high. We should have a control over the drug release. Drug release characteristics should be reproducible. And we also have to make sure that whatever drug are being incorporated into these nano formulations, they should be protected from the hostile environment of the eye. So all these features are to be included if we see a future where nanomedicines will be the predominant formulations for the treatment of the ocular diseases. Now, there are significant barriers are existing for the any formulations to cross the target sites in the eye, and the nano formulations offers benefits because it can successfully cross these barriers which are existing at the various regions. For example, if we are using the nano formulations, we can achieve penetration through the protective mucins which are existing in the tear film. Tear film has several layers, and the innermost layer comprises of the mucin. So any drug has to cross that mucin, and when we are using these nano carriers are better able to cross the protective mucins. So what benefit we will be having is we can have enhanced residence time of these formulations, and also the penetration will also be increased. Another approach by which this nanotechnology can be useful is we can also enhance the penetration and duration in the ocular surface tissue, because in this way, what some for anterior segment diseases and specifically for the ocular surface diseases, these nanocarriers we can enhance the penetration and we can increase the duration for which the carrier is available at the ocular surface, and from where it can release the drug in a controlled manner. And also it is important because through these nanocarriers, they can also penetrate through the cornea. They can also, because for the diseases like glaucoma or for the intraocular inflammation, it will be sufficient if the drug is able to cross the cornea and it can reach into the anterior chamber. So this nano carriers also offers this advantage of crossing this barrier. And it also holds promise for many gene delivery, because now there are the concepts of gene therapy is coming up, and there is immense scope for the carriers to deliver these genes, because ultimately for number of eye diseases we can anticipate that there will be improved treatment for these diseases. And specifically when we want to target the drugs to the posterior segment of the eye, so these nanocarriers, they are able to cross the various ocular barriers and reach to the back of the eye. And we can certainly look forward to better therapies for the various retinal diseases, whether it is diabetic retinopathy or it is macular degeneration. So in this way, the nanotechnology has all the potential to overcome the various barriers which are existing in the eye. So if we see what are the pros or how these nanotechnology can be useful for the ocular delivery applications, one is by improving the bioavailability, they can increase the solubility, and they can increase the penetration of the lipophilic molecules into the different layers of the eye. And they also have the options. We can have so many biopolymers we are having at our disposal. So we can select suitable biocompatible or biodegradable polymers according to their features, according to their properties, and we can have a biocompatible formulation. And also the another main feature of these nano formulations is that we have tremendous flexibility at our hand. Tremendous flexibility in the term of we can modify the surface charge, we can alter the type of surfactant that we are using, we can play with the concentration of the surfactants, we have a wide choice of polymers. According to that, we can modify the properties of the formulations. And all these choices we are having, that have to be tailored based on the solubility of the drugs. And controlled release is also one of the important objectives, because if we want to reduce the frequency of administration, if we want to improve the patient compliance, we will have to look for approaches where we can achieve controlled drug release. And there are also we can have to go for certain encapsulation methods where there is minimal of ocular irritation, or there is the ocular tissues are exposed to lower drug concentrations, because at higher drug concentrations the incidences of toxicity increases with conventional formulations. So when we are looking forward to these novel systems, we can ensure that at one point of time the concentrations of the drug that is exposed is relatively low. And we all these things are ultimately going to improve the efficacy of the drug for various segment of diseases. But there are also certain limitations are also there. It is not all a rosy picture when we are talking about nanotechnology, because we have to accept and understand that the nano formulations, they can be antigenic, they can have toxicities, there can be significant side effects also, because there are so many variables are there in terms of size, or whether it is surface properties, the surface charge and the hydrophobicity, and all these uh, factors can contribute to the toxicity of the nano formulations. And also one very important thing to understand is there is also a lack of standardized protocols for studying the in vivo tolerance test for the ophthalmic products, with specifically the nano formulations. So there is also immense scope for developing newer in vivo protocols for studying the toxicity profile of nano formulations. And sometimes because the whatever animal models that we are using to establish to safety of these products or for studying the immunotoxicity, sometimes these models, they may fail to accurately predict the safety of these products in human subjects. So sometimes in these correlation between these studies may not be very good, or that may be poor correlation between these studies. And also the presence of surfactants, that is also a major concern, because the presence of surfactants in the formulations that can uh, give a stickiness to the formulations. They can also contribute to the blurred vision on the eye. And ultimately, all these factors, if it is not convenient from the point of view of the patient, that is going to adversely affect the patient compliance. And also, there are so many choices of polymers and excipients. The toxicity profile also need to be established. So all apart from these limitations, there is also limitation in the sense sometimes the formulations, even though the formulations are giving very encouraging results in labs, but whenever we are going for technology transfer, we have to take into account whether these formulations can be produced in large scale. What is that translational ability? Whether we can have a control on the particle size, because particle size in nano formulations is very crucial, and the having a strict control on the particle size is a challenging disposition. And also the stability of these products, stability of nano formulations is also a concern, because over a period of time during storage, the particles can tend to aggregate. They can also, there are also chances that the drug that has been encapsulated, that can be expelled, and all these things can lead to possible systemic toxicity. And another major concern is any ophthalmic formulation that has to be sterile, that has to be pyrogen free. So whatever formulations that we are designing, all the formulations has to be sterile in nature. So either we have to make sure whether they are uh, able to withstand the autoclaving cycle, or otherwise we may have to opt for aseptic production. Sometimes whatever methods that we are using, they may not be adaptable to aseptic processing. So all these concerns have to be taken into account whenever we are going for the translational aspect of the products that are being developed on the lab scale. And sometimes there is also lack of standard protocols, regulatory approval protocols, because these nano formulations, they have certain unique characteristics and unique standards are to be there. So there is a lack of standards for regulatory approval of these set of products. And also toxicity concerns are also there, because these biomaterials that we are using as nano formulations, they may remain in the body, they may remain in particular tissues for a longer period of time. So this requires stringent evaluation from the point of view of the regulatory bodies. So number of novel carriers are being developed, are being explored for the ocular diseases. All these formulations, they are also used for being explored for other diseases also. And now for the ocular diseases also, these formulations are being explored, whether we talk about nanosuspensions or liposomes, neosomes and viscomes, cubosomes, microsomes, then microemulsions, dendrimers. then these nanomycels, nanoparticles, solid lipid nanoparticles, and also some newer concepts are coming up where the nanoparticles are being now loaded into contact lenses. And certain hydrogel systems are also being developed where the products are in the uh, drop form or the solution form. But as soon as you they instill them into the eye, they are they are transitioning into the gel form. So these are the in situ gels. So you have both the benefit of the ocular drops because it is in the drop form while we are instilling it. But once they are inside the eye, they are converted into the gel form, and this gel transition may be triggered due to the change in temperature of the eye or due to the pH or due to the ionic concentration in the eye. So in this way, the retention of these formulations can be increased. So there are so many different carbon nanotubes are also being explored, and during last few years, our research group we have been working on certain nano formulations specifically for the ocular diseases, and we have worked or developed certain nano suspensions for some antimicrobial drugs. We also have developed certain Nutrajit RL nanoparticles for ocular drug delivery, and also for the lotoprednol etabanate also we have developed certain PLGA nanoparticles. So this work is on initial stages, and we are looking forward to further um, translating this work on the preclinical stages. And one very important or very interesting approach that is gaining traction these days is the nanoparticles loaded into the contact lenses. So these are basically systems where the drug is incorporated into the contact lenses. So these are contact lenses where the drug is being introduced. So sometimes these drugs are can be incorporated as simple drug particles, or the drug can be loaded into nanoparticles, and then the lens contact lens are impregnated with these drugs or nanoparticles. So the advantage of these carriers is that the retention time is increased, because with the help of contact lenses, the retention time of the formulation is increased, and the drug is slowly released from these contact lenses over a period of time. And at any point of view point time, if the therapy has to be discontinued, it simply involves the removal of the contact lenses. So these nanoparticles loaded contact lenses, here the drugs can be uh, loaded in different manners. For example, the drug can be entrapped in a solution form in the hollow cavities that can exist in the lenses form, or even the drug can be loaded into microemulsions, and then these microemulsions can be loaded into the contact lenses. And there is also the another approach is where molecular imprinting can be done in the polymeric hydrogels. And one of the simplest methods is where the contact lenses, these are drug or nanocarriers are loaded through simple surface adsorption. And there can also be a dispersion of the nanoparticles or liposomes within the contact lenses hydrogels. And the another approach that has been explored is we prepare a surfactant and drug complexes, and then these surfactant drug complexes are dispersed into the contact lenses. Or we can also coat or embed the drug polymer film in the contact lenses. And simplest method is the soaking of the drug solution onto the preformed contact lenses. So although you may not have actual control release, but it certainly helps in maximizing the residence time of the drug and the intimate contact of the drug with the corneal membrane that has also improved. And so these are some of the approaches that are being explored lately for through the uh, using a combined approach of contact lenses and the nanoparticles. Another one approach, very interesting approach, is where they are using mucus penetrating particles. So what happens is when we are using conventional particles, there is a layer of mucus is there on the eye, on below the tear film, and this mucus is continuously turnover is taking place. And if we are using simple particles, these particles are rapidly removed from the site of absorption. But some they have been reported the mucus penetrating particles. So these particles, they are able to penetrate through the tear. They are able to penetrate through the tear film mucus layer, and in this way they are better retained at the site of absorption, and much better therapeutic outcomes have been achieved with these mucus penetrating particles. And there are also a number of nano carriers are also there in the clinical trials also, where these formulations like microemulsions are being developed for dry eye, nanoparticles are being developed for cataract management, and different classes of artificial tears, including liposomal spray, are being developed for dry eye diseases. Then mucus penetrating particles loaded with loteprednol etabanate, these are being developed. So number of other, these are just some representative examples of where nanocarriers are under different stages of clinical trial. But one very important aspect which also seeks our attention is the ocular nanotoxicology. Because uh, although the nanomaterials are having immense therapeutic potential, we should also take into account that these materials also have significant toxicity by the virtue of their size. So the similar properties which improves their biological uptake also contributes to their toxicity, and that is the reason why lately now much focus is also being placed on the nanotoxicological studies, not only their to study their impact on the humans, but also how they are impacting in our environment. So, because we have to understand these nanomaterials, they have unique properties, specifically due to their quantum size effect. So, similar particles, as soon as their size reaches to the dimensions of 100 to 200 nanometers, their properties change. Chemically, their structure may remain the same, but their physical properties undergoes a significant transition. And also, as we reduce the size, the surface area to the volume ratio also increases. So, the same particles, when they are existing as larger particles, they may not be toxic. But as soon as those materials are in the nano size, their toxicity may increase manifold. So even, for example, even nanomaterials which are made of inert elements like gold, they become highly active in the nanometer dimensions. For just for some example is, for example, copper. Copper is a opaque substance, but as soon as they are in the nano size, they become transparent. Even simple elements like your platinum, which is otherwise an inert material, but as soon as you are taking it as nano dimensions, they become catalyst. The stable materials like your aluminium, they are normally stable, but they turn combustible when their size is in the nano range. Similarly, gold, which is normally a solid, but they turn into liquids at room temperature when you are using them as nanomaterials. And one very common element is the silicon, which is generally it is an insulator. But if you are using it as a in the nano dimensions, it becomes a conductors. So sometimes we have to understand this. The remedy that we are using sometimes, it becomes the cause of side effects, or it sometimes proves worse than the disease itself. So that is the reason why we have to take into consideration what are the toxicological profile of the nano materials, because there are also number of diseases are also being caused due to the nanoparticle exposure. Because for example, number of diseases are also being reported to be triggering factors for a number of ocular diseases. So it is a, you can say it is a double-edged sword. You have to use the nano carriers very cautiously, because we are using them for their beneficial purposes, but they should not be the cause of toxicity or side effects or generate newer diseases. So when we are taking into account what are the various toxicity concerns. So the nanoparticle toxicity is influenced by a number of factors. So the major factors which contributes to the toxicity of the nano formulations is first and foremost is the what is their chemical component and what is their nanoparticle chemistry. Because chemical when we are using the term chemical component basically refers to the chemical elements that the nanoparticles may be containing. So two kinds of nanoparticle, they may be having the same components, but they may be having different crystalline structure. So having a different crystalline structure drastically changes its toxicity profile. Another very significant factor is the size and shape, because as we are increasing the size of the particles, the surface area increases exponentially. So the chemical molecule may attach to the larger surface. When the size is being reduced, there is much larger surface area is available, and this larger surface reactivity surface no doubt increases its reactivity, but it also increases the toxic effects also. And for example, silicon oxide nanoparticles, which are having diameters less than 40 nm, shows a higher toxicity to human corneal epithelial cells than those bigger in 50 nanometers. So size is very crucial. We have to strike a balance between the benefit and risk ratio. That has to be the basic rule that we should not forget. And in addition to the smaller size particles, the smaller the size of the particles, they are much more able to cross the various epithelial barriers. And as they are able to migrate across the epithelial membranes, they may be the cause of cytotoxicity, or they may also induce the inflammatory responses. And size, apart from the size, shape is also very important, because shape also influences the toxicity. For example, the nanoparticles which are having higher aspect ratio tends to be more toxic. For example, the fiber shaped nanoparticles are more toxic than the spherical shape particles of the similar chemical composition. So size and shape is very crucial. Apart from that, surface area and charge, that is also very important, because when we are using the nanoparticles which are having the same mass, same chemical composition, and same crystalline structure, but the inflammatory response is proportional to the total surface area. So larger is the exposed surface area, there will be greater reactivity, there will be greater chances of oxidative and DNA damage. And as there is a change in the surface charge, it can also influences the biodistribution also. For example, the positively charged nanoparticles, they are more easily taken up by the cells as compared to the neutral or negatively charged particles. And this can lead to more inflammatory effects and cell death. So the charge also have a crucial role in the toxicity of the nanoparticles. Then dosage and concentration, and that is also very important because low dose exposure for a longer period of time, maybe sometimes more safe. But sometimes what happens is, when you are using higher concentration of nanoparticles, in higher concentration, there are chances that particles can tend to aggregate. So even though the particle drug is at higher concentration, nanoparticle is at higher concentration, but because these particles, they have aggregated, that can reduce the toxicity. So sometimes the lower concentration of the nanoparticles may be more toxic, because in the lower concentration, they are existing in separate forms. But at higher concentrations, these particles are aggregating, so they are not able to cross the biological membranes, or they are not able to exert their biological effect or toxic effects. Then the behavior, what is their behavior in the solvent or in the biological media? Because the media also have a significant role in their dispersed state, or whether they are agglomerating. So all this solvent or biological media can influence their size, their agglomeration, and have an impact on the toxicity profile. Just for example, the titanium oxide and zinc oxide nanoparticles, they show considerably bigger size in phosphate buffer saline than in water, and thereby their toxicity also changes in these different solvents. And also in these nanoparticles, they also behave differently in different biological media due to the formation of protein corona. These protein corona, these are the layers of proteins that are adhered on the surface of the nanoparticles. So when these protein corona is present over the nanoparticles, this changes the size and surface properties of the nanoparticles, and once their size changes, naturally that will affect its absorption, that will affect its transportation, and all this will have an impact on the toxicity profile of the nanoparticles. So these are all different aspects that we have to take into account while we are looking forward for designing a nano formulation. We have to, sometimes the nanotoxicological concerns are overlooked, but this is also one of the very emerging areas where the nanotoxicological profiling of the formulations are to be done when we are working on the or devising or designing certain newer formulations for the eye. So, what does the future holds for us? If we look into the future of ophthalmic products, we must understand that still now the eye drops constitute about 90% of the total ophthalmic formulations. And we all know what is the reason behind the popularity of these eye drops. So, first and foremost, target for us is to come forward for designing certain formulations for the eye which are based on eye drops. And also, we must also consider that there are stringent regulatory requirements for new ocular delivery systems, because the products are meant for one of the most sensitive sense organs, and they are to be sterile, they are to be pyrogen free, and specifically when we are taking into account the nano formulations, the various toxicity concerns are also to be emphasized upon. And we should also understand and accept that no single device or technology will be sufficient to meet the range of needs, because there are so many barriers are there, so many requirements that are to be met, and sometimes one simple strategy may not be sufficient. So we have to look forward to how we can combine the approaches, how we can integrate the multiple approaches to optimize the drug delivery to the eye. So we have to look forward to integrating all these approaches if we want a product which can be effective in treating the diseases, whether it is anterior eye disease or the posterior segment diseases. So, I would like to end with this simple and profound quote that if you are looking at the big picture, you have to think small, really small, about how you can improve the things. And it is here that the nano carriers have a significant role, especially when we are looking forward to designing a product for treating the ophthalmic diseases. So this is the research group of mine who have been pastly working on different aspects of the ocular formulations. And thank you very much for patient listening. And I hope I have been able to generate some interest in the participants who are registered for this webinar on the various opportunities that are existing for designing products for ophthalmic diseases. So that is all from my end. Thank you all for your kind attention.
Thank you, ma'am, for sharing such an insightful and thought-provoking lecture on eyeing the future, platforms for enhanced ocular therapeutics. Your expertise in nano carrier systems and advanced drug delivery strategies has given us invaluable perspective on the future of the ocular therapeutics, and we are deeply inspired by the innovative approaches you have presented in today's webinar. Your dedication to advancing pharmaceutical sciences and improving patient care shines through in your research. And we are fortunate to have had this opportunity to learn from you, ma'am. Thank you once again for this enriching session and for contributing your time and knowledge to our 72nd SPSR webinar. Ma'am, we have some questions. I take them one by one. So the first one question is, Could you share any recent breakthrough in gene therapy or cell-based therapies for ocular diseases?
Recent cell-based therapy, I should admit I'm not in a position now to share with it, but there is significant research is going on at preclinical levels, and because there are number of promises are there for gene therapy, and the only challenge is how to deliver that. So at preclinical stages, it is going on with liposomes. Liposomes are one of the carriers which are widely being explored for the ocular diseases, specifically for targeting through the posterior segment diseases for large molecules.
Ma'am, we have another question. How can ocular drug delivery platform minimize side effects and improve patient compliance in chronic eye conditions?
Okay, so like one of the major constraints with the conventional formulations that are there for chronic conditions, whether it is glaucoma, that is one of the most prevalent ocular diseases, is the frequent administration, the frequency of administration. And these are drugs because eye has an inherent defense mechanism where as soon as you are instilling a drop, the tear secretion starts. So there is rapid washing out of the formulations from the eye, and there is very limited time that is available for the drug to cross the biological membranes. And due to this naso uh, tear secretion, there is this drains through the nasolacrimal tract. And this, once the drug is drained through this nasolacrimal tract, the absorption of the drugs takes place, and this is responsible for the systemic side effects. So, whenever we are looking forward to certain novel formulations which can be retained in the eye, where the drug is released in a constant manner, this can minimize the systemic exposure of the drug. It will provide a platform or a system which is retained at the site of absorption for a prolonged period of time. So, no doubt this will reduce the frequency of administration, the patient convenience and compliance will increase, and all this ultimately translates to better therapeutic outcomes. I hope I am able to answer to the question.
Yes, ma'am. Ma'am, we have another question. In what ways can ocular delivery platforms be tailored for personalized medicine, considering individual variation in eye anatomy and disease?
Because these formulations, as per the prevailing condition of the patient, it gives us flexibility for the dose modification. They can be uh, because formulations that you can instill into an eye, that cannot be more than one drop. If you instill, instead of one drop, if you instill two drops, that will be drained off. So within that one drop, we have the flexibility of changing the or modulating the amount of drug that is required for that particular patient. So that gives us an opportunity, and when we are looking forward to small scale preparation, so that also gives us the flexibility also.
Okay, ma'am. Ma'am, we have one more question. Can you discuss the future potential of the implantable ocular devices for long-term controlled drug release?
Yes, there is a significant market is there for the implants also, because once you are implanting it, it is a one-time affair where that can be implanted. For example, for diabetic, especially when you are talking about the posterior segment diseases, where still it is a challenge for us to design a system which can cross the biological membrane. So here the implants offer significant advantage, and some products are already there in the market for the as the intravitreal implants.
Yeah, okay, ma'am. We have some more questions, but I think you have already answered them in your presentation. Uh, so thank you so much, ma'am, for answering all the questions so thoughtfully and thoroughly. And your clear explanations and insightful responses have enriched our understanding of advanced ocular therapeutics and inspired us to think about the future possibilities in this field. We truly appreciate the time you have taken to share your knowledge and expertise with us today. Thank you, ma'am. So, ma'am, now I want to conclude the session. As we conclude our 72nd SPSR webinar on eyeing the future platforms for enhanced ocular therapeutics, I want to extend our heartfelt thanks to Ma'am Professor Preeti K. Suresh for leading us through an enlightening session. Today's discussions have highlighted the incredible potential of the advanced ocular drug delivery platforms, from nano carriers and polymer-based systems to innovative non-invasive methods, all aimed at overcoming the unique challenges in treating ocular diseases. We have seen how these emerging platforms are not only improving drug stability and bioavailability, but also opening doors to more personalized and patient-friendly treatments. The insights shared today by ma'am encourage us to think boldly about the future of the ocular therapeutics, where technology and innovation continue to enhance patient outcomes and quality of their life. Uh, we are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all SPSR team members and participants for their collaborative effort, which have broadened our understanding and fostering cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continue collaboration in advancing pharmaceutical sciences and research. We are grateful for your significant involvement in the 72nd SPSR webinar. Your engaged participation and input have been essential, and we eagerly anticipate collaborating on upcoming initiatives aimed at enhancing healthcare and well-being for everyone. Once again, thank you, our today's esteemed speaker, Preeti ma'am, and all the attendees for making this event a success. We look forward to your continued engagement in future discussions. Stay connected, stay innovative, and together let's continue to enhance healthcare outcomes for all. Our upcoming 73rd SPSR webinar is titled on Pro-Drug Strategies for Colon Targeted Drug Delivery. Our esteemed speaker for this event is Professor Dr. Sunila Dhaneshwar, Director, Amity Institute of the Pharmacy, Amity University, Maharashtra, Mumbai, India. So this is all about. So thank you, ma'am. Thank you, all the participants. Thank you so much.
Thank you everyone for this opportunity, especially Dr. Shashi Alok, Dr. Monika Sabharwal, and Nidhi Mittal for your active involvement and very smooth conduct of this webinar. Thank you very much.
Thank you, ma'am.
Thank you. So I can disconnect right now. Thank you.
Yes, ma'am, you can disconnect. Yes, ma'am.